All right, so it's my pleasure to introduce Lewis Wheaton. Many of us know him already. Lewis has been a real champion for neuroscience here at Georgia Tech, leading both some really cool research initiatives, and most recently, in fact, today he is in the process of submitting a major developmental grant for neurosciences here at Georgia Tech, with both a focus on new research directions and a focus on increasing diversity, equity, and inclusion in neurosciences. So he's done some really championship work on that. I think it's very impressive that he's giving us a seminar the same day that this huge many $1 million NIH grant is due. So we're delighted to have him. What we get to hear from today, though, is Lewis's really incredible research, and I'm really excited to sort of get new insights into the direction that his lab is going. Like a lot of us here, he has interest in aspects of motor neuroscience, but sort of on a very different scale than like what I and some folks do, focusing on skilled movements, especially in the upper extremities, and on rehabilitation. He came to Georgia Tech in 2008 after he had his PhD at the University of Maryland, and worked with the NIH. And since then, he's been directing his lab here and doing some really great work. He also is affiliated with the Department of Rehabilitation at Emory, as well as being part of our biological sciences department. Lewis, it's a real great pleasure to have you. We're looking forward to your talk, and I'll be collecting questions on chat like we've been doing in the previous weeks, and I'll facilitate those at the end. And I'd really like today to have the first question come from a graduate student or a postdoc. So I'll be asking for a graduate student or a postdoc to ask the first question. All right, Lewis, go ahead and take it away.
Thanks, Simon, and great to be with you all today, albeit in this virtual space. Good to always talk science as well. So today I'll be giving a bit of a summary, kind of a bit of a historical summary, but also kind of leading up to some of the work that we're endeavoring, looking at this very well-known network, these parietofrontal networks that have been really well established and well characterized in humans in understanding motor control and motor planning. I always try to make sure people know that all of my work is in humans. I don't do cellular or non-human primate or any other model system. All of our work focuses on humans and really trying to span basic science and translate those things into motor rehab principles. And so today we'll be talking a good bit about that. One of the things that's really interesting when you go back, and I'm always a fan of the history of the field of neuroscience, is when you go back to a fairly recent history, really in the 80s into the 90s, you see this explosion of studies that are really driven by some clinical questions. Where researchers, and some of these researchers, if you're in the field of upper extremity motor control or motor control in general, many of these names will ring a bell. These were people that were back in their postdoctoral and in some cases grad student days doing imaging studies where they were, in this case here, this is very classic work where using PET imaging approaches looked at areas of the brain that seemingly were involved in motor imagery. The impetus of this research was really focused on clinical questions that were trying to understand where in the brain are people having, or do people internally plan motor control relative to injuries that can result in motor planning deficits. And one of the principal findings of a lot of these studies, and this is a pretty common finding, is that particularly over the left hemisphere in motor and premotor areas, particularly on the more ventral surface, you see these activation patterns. And related work on the other side of the screen is a very classic study by Alex Martin, Leslie Ungerleider, James Axby, where they were really interested in very similar brain areas, in this case here, looking at the role of tool use and recognizing that there were certain parts of the brain, particularly again in some of these same areas, particularly in the premotor cortex in the left hemisphere, that were also active for tool use. And even in this paper, they reference this other work here indicating that, yeah, the same area that was active for motor imagery of tool use is also engaged in this knowledge, or I'm sorry, the same areas that are involved in motor imagery are also involved in tool use. And this idea really started to blend together of that, maybe these left frontal areas and as well, including into the parietal lobe, are very active in the ability to encode motor skills in human behavior. That segued beautifully into the explosion of what you might refer to sometimes as mirror neurons. These neurons in various parts of the brain, as is indicated here, in the parietal lobe, particularly in the area that we sometimes refer to as the posterior parietal cortex. If you do invasive recording here and in this monkey in this region, but you can do the similar things in frontal areas, particularly around the arcuate sulcus and the spur of the arcuate sulcus in the ventral premotor cortex. You can find these neurons that are active when a monkey either performs or observes the performance of certain grasping or motor control or object-related or tool-related behaviors, right? And so in this case, this monkey reaches and grasps to an object with their hand, and what you see here is this activity before the reach to grasp, and similarly here, before extending the arm to pick up the piece of food. And so this idea here is that not only can these same areas be involved in the actual performance of the task, but even in the observation. And so if you were to characterize a bunch of these neurons in monkey 1 and monkey 2, you would see various populations of neurons that are involved in certain behaviors, right? Whether it's object-based, biological motion-driven, space-driven, or things like that. And so this kind of generally led to this idea of the importance of the mirror neuron system. And in connecting this primate work to the human work that was really bubbling up at the time, you see this explosion, really, of studies in human neuroimaging that link to parietal frontal areas and their relationship to upper limb motor control. And so much so that it's almost become the default thing to find. Even in some of our studies, if I were to just kind of go through some of our prior work, whether we're using non-invasive or invasive methods to understand human brain, understanding action judgments usually involves left parietal frontal systems, whether it's motor predictions with tools. These are a different slice, but again, you see frontal and parietal areas heavily involved in this task. In this particular case, there's left hemispheric bias. Similarly, in some of our older work, the graspability of tools, tool selection, and even using intracranial recordings, we've shown that reach to grasp of tools and objects shows those similar patterns of neural activation when you're reaching particularly with the contralateral limb as opposed to the ipsilateral limb, or pretending to do just shoulder movements or just simple wrist movements that aren't reaching movements. So these left parietal frontal systems and these activations abound, and we've been able to sort of, in these studies and even other studies that I haven't talked about, made some, casual inferences about what's going on. But this is a problem that all of us in human neuroscience deal with, because we really don't have a strong idea still what this activity really represents. It's really hard to nail down exactly what's going on in these systems. And so one of the things that has been kind of an overall focus and interest of mine over the years is taking a step back and looking at the question of what's going on in these systems using multiple approaches. And really, the centerpiece may still be neurophysiological and neuroanatomical approaches that we commonly use in human brain imaging, such as electroencephalography, where we're capturing voltage-related information from the scalp, or we're doing functional magnetic resonance imaging, where we're looking at changes in oxygenated and deoxygenated blood flow in various parts of the brain. But we also want to understand how these neural changes relate to certain biomechanical findings, and also even computational theories about how motor control works, and try to leverage tools and paradigms to connect all these things, right? And to see if we can really get a better sense as to what's going on, particularly in these parietal frontal circuits, if we're looking at the problem kind of from both directions. So today I'm going to talk about three themes, not necessarily individual studies, but three themes here of work that have gone on in the lab over the last few years to try to understand aspects of motor control related to how we think about what's happening within the parietal frontal system. We'll look at the first, my former grad student, Nick, who's at UC, good grief, I just blanked on the city name, but he's in California now, doing some good work out there, looking at prediction of judgments of action goals. Bill Cusack, who is now at Facebook, actually, doing some really cool work up there, and his work looking at perception of action coupling for motor learning, and Crystal Topping, who's really done some beautiful work on perception action related to skilled learning and skilled action development. So to really understand what's going on and to almost explain where this is going before we get started, well, we're going to think about the idea of eye movements. Eye movements have been one of those things that have been really well studied over the years when it comes to motor control, and particularly motor control related to the upper limb. This is a very classic study, and I don't want to get too deep into the weeds on what this study and studies like it have tried to take a look at. But one of the things here we see with this particular work, when you have to saccade back and forth between targets, and then subsequently you have to reach to a target and work your way back without visual feedback, that your eyes tend to predict basically the future direction of your hand, right? And so the idea here, to put it in a very crude sense, is that your eye is sort of leading your hand through time and space, right? And that, as you're reaching, you're engaging the ballistic reach, your eyes kind of stepwise advance where you're going to go. And they primarily are in line with the direction of your reach as well. All right, so most of the reach displacement, your eyes tend to go in the same displacement of that reach. And so we wanted to understand or use eye movements as a window into understanding certain things about motor skills and tool use and tool observation. And that really led to some thoughts about what is going on potentially within the parietal frontal system. So I'll start with a very simple behavioral paper. This is a behavioral study that was done in collaboration with Anna Borgi in Rome, who was really interested in doing some studies to understand certain aspects of tool use and tool use context. And what we did in this work was we started with a very basic image where we asked people, here's an image, and this is an example. If you can't quite see it, it's just a spoon or a scooping spoon, and there's like a tub of ice cream here. And you're asked to say, is this tool and object pairing correct, right? Very simple. And that wasn't the only image, obviously. We showed other images. In this case, there was a hand in the picture and a functional grasp, meaning that the grasp suggested that you were going to actually use the handle for the purposes of scooping out the ice cream, or what we call a manipulative grasp in this case, where you're grasping the bowl end of the spoon, which is not very helpful for scooping out ice cream. You'll make a mess that way. So this is kind of a interfering grasp to the overall tool usage. Similarly, we vary this context a couple different ways, where we also put it in an incorrect context in a way that doesn't afford natural action. In this case here, there's a tennis ball in the scene. And in a spatial sense, if you can't quite see this, these are sugar packets. And so it's not uncommon to see, let's say, sugar packets and spoons in the same setting. You don't necessarily use them together, but they're very commonly associated with each other. And so the task is simply to evaluate if the tool-object relationship is correct. The important part about this is the hand is immaterial to that judgment, right? It has no bearing on the judgment of the usability of the tool on that object in the scene. And so the findings behaviorally, we looked at reaction time, and we looked at reaction time two ways. Reaction time with the hands, how long does it take for you to press a button to determine whether or not whether or not the context of the image is correct. And as well, we had people, a separate group of people, make the same, perform the same task with their feet. All right, and so in that case, the foot switch reaction, and the idea, the motivation behind that is if something is perhaps confounding for the hands, there is a sense that behavior with the hands can be affected, but behavior with the feet is actually not affected. And that basic thing is what we found. There were only latency effects when they were hand-driven, but not when they were driven by the foot. But what was the basic reaction? The basic result suggested that when objects particularly were shown in a spatial context, decisions about correctness or incorrectness were always longer than when they were in the more appropriate functional context. You're able to make those decisions a lot quicker. Likewise, importantly, the manipulative grasp posture, so the posture in this case here, grabbing the coffee pot by some other space than the handle, which is very atypically done, that tends to elongate reaction times in these individuals. And again, the hand has nothing to do with the decision. That's a superficial artifact that has nothing to do with the actual behavior. Forgive me if you hear a dog barking in the background. Dog rivals abound in our neighborhood. So, and this really was fascinating to us, and it led us to really think about what's driving this behavior some. And what's exciting is that the gut of this experiment is really based on how you appreciate the visual stimulus. And so the natural thought for us was to look at, well, let's look at vision. So if we take the same experimental structure and let's add eye tracking and see what happens as people are looking at these same stimuli, and now we're inferring something about their gaze. And so here is kind of the model of how we looked at this. We kind of separated out each one of our tool object pairings based on looking at areas of interest. In this case, here's the object. And then we looked at both ends of the tool, and we also included factors like the hand, the arm, and even null space. We modeled that in, but really for the purposes of the reporting and the paper, because those other null spaces didn't really matter so much statistically, we really modeled out their gaze patterns by looking at not only gaze within an AOI, so gaze shifts around a single area of interest, but also any combination of those three, or at one point we had even up to 10 potential areas of interest that we were looking at. We really pruned down to the three in a data-driven way. So this is a characterization of what you can see if you were looking at particularly this object and we were looking at the hit count, basically, of being in any one space. This is a typical gaze profile for most people. They would start out at the object, and then there would be a drop-off where they would hit the manipulative tool in, which is this end up here, where, again, this is the thing that is going to operate on the object. And then they would drop to the functional, then back up to the manipulative, and then swing over to the object. So this is what we really cared about. We didn't care so much about the latency and the onset and the duration. We cared more about their patterns. What were their gaze patterns, and was there anything interesting about the patterns that was ultimately predictive about behavior? And so if we were to track their probability over time, looking at each one of these three spaces, in the correct no-hand condition, we see here that people tend to, as we almost expected, they look at the object first, and that probability drops off, and then quickly their probability goes up here at looking at the manipulative end. And remember, the manipulative end is up here. So basically, in this case, their eyes are going back and forth between these two features, right? And the handle, which is this black structure, this black line here, really never comes as a point of interest at all. And so if we were to mark this out using this trajectory space here, we're able to describe the bias, the foveal bias for each area of interest for each condition set. So for no hand, in the correct context, we see there's a bias away from the handle but more towards the object and the kind of operant end of that object, the manipulative tool end. This kind of space here is a point of equivalence statistically, where there's equal probability in being in any one space at any time. Now, in the functional context, so again here, the person is grabbing, looks like they're grabbing the handle, and again, importantly, the only decision you have to make is whether or not the tool and the object are aligned together. And what we see is gaze immediately starts to shift down to follow where the hand is, right? And so it biases a little bit more towards that end, getting a little bit closer to equivalency between all areas of interest. And in the manipulative space, it swings right back, and now most of the bias is towards the manipulative end instead of the object, but there's very little attention really ever driven to the handle. This is actually very important for us because a lot of studies related to action encoding and mirror neurons have always suggested a real importance of tool-object and tool-object interactions of seeing the hand engage with the tool. And the fact that we did not see this as a principal area of interest in any of our studies was actually very intriguing, and a point that I'll come back to here in a moment. Now, we did a hierarchical clustering analysis of this, and one of the things that's really kind of fascinating, I'll just quickly point out, is that really the type of grasp is what separates out the cluster. So if you have a manipulative grasp, a no-hand grasp, or a functional grasp, these all have a heavy preponderance of clustering out these different categories of gaze when we look at the scale Mahalanobis distance for each one of these contexts. Now, what we wanted to build upon here was, well, let's think about this from a neural point of view. Now, let's add in some of the brain imaging to think, okay, now that we understand something about the fact that their gaze is different depending on which object or which scene they're looking at, can this tell us something about the underlying parietofrontal circuitry that we're seeing? And so we designed a study where we actually just looked at the no-hand functional manipulative grasp conditions, looking at correct, incorrect, and spatial tool use, right? And so these kind of 3x3 grid of particular tools. And we modeled this study in two ways. In the first experiment, participants really weren't able to see the image long enough to make a saccade. So that was a 100 millisecond experiment. So the image really flashed up for 100 milliseconds and it went away. And we posited that we would be able to inhibit saccades during that condition, but that it would not affect their actual behavior. Similarly, in experiment 2, the stimulus was on the screen for 500 milliseconds, long enough to make 2, maybe 3 saccades to evaluate the areas of interest. What's really nice about this is that when we look at their behavior, because we do ask them, was the tool-object pair correct or incorrect, there was equivalent accuracy regardless of the 100 or 500 millisecond experiment. So somehow using peripheral vision, they were able to capture enough information to make the decision in the 100 millisecond time interval. And we know this because we can look at their vertical viewing angles here and note here in the 100 millisecond experiment, most of their fixations and saccades are lined up in this null space where there was a fixation cross. So we can verify that, yeah, there was no gaze that really shifted away from that. But in the 500 millisecond experiment, we can see that basically they would go back and forth as quickly as they could between the object and the tool. Importantly as well, this upper area is where that manipulative end is, and the handle is always down here. And you see most of the gaze is kind of going in this upward trajectory. We'll quantify that here in a moment. As well, we could also see that, most people, they could make a saccade within about, 100, 200 milliseconds. And this 500 millisecond time interval is enough to make a first and perhaps a second saccade. The saccades tended to always go to the object and then towards the tool, still verifying what we had seen before. Now, I would normally show what I'm about to show in a video, but I can't because I don't want to have problems with video recordings on this platform. But I'm gonna kill you with head maps here. And so I will explain these in another image, but I just want you to see what we're able to see and where the next data that I'm going to show you comes from. So in the 100 millisecond experiment, we're able to look at these are every 10 milliseconds, starting at 30 milliseconds after the stimulus comes on, and we're able to kind of look at 10 millisecond steps all the way through the experiment. For the 500 millisecond experiment, we're able to do the same thing. Again, these are maybe the first 3 right after the stimulus shows up, and then we kind of fast forward time a little bit to 355 and on, remembering the fact that the stimulus is still on the screen for the 500 millisecond experiment, but it has gone away for the 100 millisecond experiment. So what's the takeaway here? What we really wanted to see here was, is there some vulnerability of these parietal and frontal areas to be able to evaluate these tool object images based on this structure, based on the experiment that we set up. And so when we look at, for example, the role that grasp plays, what we see, and this is a different representation, we've taken the significant activity of each electrode over time and lined it up. And so here what we have is the frontal, or the lower numbers, and then you work your way back to the central electrodes, and the parietal occipital electrodes are here in the higher numbers. So it's basically frontal to posterior, lower number to higher number in rank order. And what we have here is the main effective hand. So with a grasp type, really you see that activity coming in at about, maybe about 150, 200 milliseconds after the stimulus onset. In the 500 millisecond experiment, we see these 3 phases of activity. And basically, to summarize, it goes from parietal to frontal to parietal, right? And it kind of coincides with the saccades, which was kind of fascinating to us. And so my first question to Nick really was, is this some kind of funky artifact here? What's really happening here? Because usually with these artifact can be a big problem. And so, and these kind of, to answer that question on one level, we said, all right, in the 500 millisecond experiment, what are the condition-based differences? And what we see here is that particularly over frontal areas, the manipulative grasp condition has tremendously more activation compared to the no hand condition. And also there's some subtle condition differences as well in the parietal lobe, but the frontal lobe in particular is where we found most of the effects. And so now I still have this question. Is there some weird eye movement pattern that may be being picked up as an artifact in the signal that we're not accounting for? And so now if we look at it through the lens of context, so it's correct, incorrect, or spatial, what we find here is that regardless of it being the 100 or the 500 millisecond experiment, we basically see the same activation coming up over frontal and mid-parietal and parietal areas around the same time, around that 500 millisecond time period. And so this suggests to us that the effect that we're seeing over here, it couldn't be driven by an eye movement artifact, because when we sort the data just in a different way, that if it was an artifact, it would still be robust in this consideration. But what we're really seeing here is this modulation of that parietal frontal activity, in particular that frontal activity, when people are engaged in understanding this grasp-related behavior. So whether or not it's manipulation grasp or functional grasp, seems distinct from understanding the direct or the distinct tool use properties. As well, it seems as though the frontal activity may be related to understanding something about strategy or intent of the behavior that is going on. And so this kind of dual representation of perception and action coupling and action observation really got us excited and got us thinking about some things related to rehab in the rehab space. And so I'll transition now to think about these studies from the rehab space and really realizing that a lot of motor rehab does focus quite a bit on the idea of action observation. Indeed, there's even a brand of rehab called action observation-based therapy, where individuals can, in this case here, you can stare at a screen or stare at another person in real life, performing an action, and the intent is that that visualization of that action will help you. There are plenty of studies that suggest that action observation alone or action observation along with motor imagery are helpful and robust to teaching people how to perform motor control tasks in certain settings, whether they are trying to recover from a stroke or in recovering motor control after a stroke, but also even speech. There's a lot of work in this with speech therapy to demonstrate or visualize how to develop speech sounds, and that visualization can then help infer something about how these people should be performing their speech motor control. Now, in our case, one of the things that we're particularly interested in is upper limb amputation. Upper limb amputation is fascinating because it changes not only body morphometry, so now you're missing part of a limb, you also have lost the sensory feedback from that limb. As well, a huge component of reestablishing motor control tends to involve the use of prosthetic limbs, artificial limbs. And so we started to wonder, is there something unique or unique opportunity for action observation in the situation where somebody has an upper limb amputation? In some of our previous work, we've actually suggested that the visual system may actually be very well positioned to support motor learning after upper extremity loss. In a small cohort of individuals, we recorded their neural activity using electroencephalography while they basically performed a targeted reaching movement with their upper limb. And we compared this with individuals that were sound limbs that were either right or left hand dominant. All of the amputees in this particular population were formerly right-handed dominant, and they all lost their right limb. And so we wanted to see really if there was a switching of their functional dominance as it relates to this, but that question quickly got supplanted based on the results that we found. And the results are pretty straightforward. Effectively here, if you're a right-handed person performing a task with your right arm, we see most of the activation is in the left motor cortex, very expected. If you do something with your left arm and you're right-handed, not an amputee, but you're left-handed, you're also going to do things, and motor planning is going to involve both hemispheres. That's very much expected. Likewise, that pattern switches if you're left-hand dominant. If you're left-hand dominant, you tend to, if you're moving your right arm, you tend to engage more bilateral activation motor planning, but when you're moving your dominant arm, you tend to engage in more contralateral motor planning behavior. But what we saw in our amputees really kind of stunned us a little bit. These formerly right-hand dominant amputees, when they move their left arm, they show motor planning activity that looks much more like left-handed dominant individuals. But when we look at their amputees, a lot more motor planning involved in posterior parietal brain areas, parietal occipital areas in particular. So that led a lot of work that we've been doing over the years to really harness the visual system to promote these functional improvements. Is there a way that we can actually do that? And through many studies that we've done in the lab, as you see there, we modeled this idea to really use action observation therapy and to really focus in on this idea of strategy. I mentioned that before. Are there strategic benefits to really being able to understand or see how another prosthesis user is engaged in motor tasks, because it is so different from a biological limb? And so in this case here, we created these two conditions where somebody using a prosthesis watches somebody engaged in biological limb imitation, which is mismatched, versus prosthesis imitation, which for an amputee would be the matched limb training. And the summary of this is that there is a big difference between the two. And effectively, the prosthesis user, if they are watching another prosthesis user doing the task, they basically mirror exactly what they see. In this case, this would be what the prosthesis user sees. And what this is really, without getting into a lot of the nuance here, this is just an angle representation of the relationship of the shoulder and the elbow moving together in time with a task. And so for somebody with sound limbs, which is in gray, that you would see kind of this shape. Don't worry about what that shape represents. And for prosthesis actor, you would kind of see this shape, this relationship. So a different slope between those two. What the participants do if they are a prosthesis, if they're imitating another prosthesis user, their angle-angle relationships between the shoulder and elbow begin to look a lot more like the prosthesis actor. However, when they're trying to imitate an intact limb, they're still going to try to perform the action the same way the intact person does, which, as we see in a lot of our studies, is highly detrimental. They actually perform very poorly when we look at biomechanical and other behavioral constraints. And so this really got us excited, and we wanted to dig into a little bit more as to why. What's going on within this structure that allows this to happen? And so a group of students, in this case led by Crystal, really wanted to understand the role for the visual system in supporting this matched limb training. Again, if you're looking at something, there's probably a visual pattern to how you're looking at it. And so what we did in this study was we took prosthesis users and we assigned them to one of two groups, either matched limb observation or mismatched limb observation. And in this case here, they watched a user take a disc, move it over a barrier, and place it on the other side. Very simple, very boring task, right? And then once they watched it, they were also then supposed to perform the action. And they did this in 3 different blocks, right? So you observe for a little while, and then you perform the task. And then you observe for a little while, and then you perform the task. You observe for a little while and perform the task, depending on which group you're in. And one of the things that was really exciting for us when we started to look at the gaze patterns, we had several areas of interest. We were interested in how they use their shoulders. The shoulders are very important for this particular task because really the shoulders, or flexion of the shoulder, is what controls the actuation of the prosthetic limb. So if you flex the shoulder, it pulls a cable system. You can kind of see the cable here. It pulls this cable and it causes the prosthetic limb to open or close. This is a very specific skill that takes a good bit of time to actually learn how to do and control, and is incredibly unique to prosthesis learning because if you're doing this with your hand, you will probably not use your shoulders very much for this particular task. We were also interested in looking at visually, where do their eyes go? You have to be able to clear this barrier, so we're interested in their ability to kind of watch the gaze clearing the barrier. But there's an alternative where your eyes can basically just go between the two targets, between the start and the end points of the action. And to summarize, what we found is here. In this case, for the matched limb users, their eyes really clear that barrier and go back between the start and end points of the action. However, in the mismatch condition, they're really just going back and forth through the board. Gaze through the board, which is actually not possible biomechanically. You can't move the disc through the board. And so the fact that they're observing the action crossing the board suggests that they're picking up on some subtleties of the movement in action observation that may be helpful in controlling their behavior. The question is that true? Well, when we look at their kinematic behaviors, we're interested particularly in a couple things. Remember, they have to go over this barrier. And one of the things that we were interested in is looking at their peak height. So this is basically, as I've kind of called it before, it's like an estimate of risk. How close are you willing to get to that barrier to make your grasp and the movement efficient? And you notice here that mismatch users, they basically have the same peak height across tasks between trials. However, the matched group, they tend to get a lot lower, and they have a significant decrease from trial 1 and 2 to trial 3. A good sign for optimizing movement, as well as the fact that while their peak height is dropping, their velocity is going up. And so they're very interested in not only engaging in the riskier height, but they're also happy to move faster as they're employing that behavior. Likewise, we also see a subtle decrease in the amount of peak lateral trunk movement in the match group, which is also quite fascinating. Uh, as well, in continued analysis that we're looking at here, particularly looking at their shoulders, their ability to look at the shoulders. It seems like their shoulder, or the probability of them looking at their shoulders, may correlate with their peak velocity. So the more you're looking at, the higher the probability that you're going to look at those shoulders, which may help confidence while you're making the movement and while you're moving your body through this task. It may correlate to improvements in that peak velocity to help support motor control and motor learning. But I haven't talked much about the neural side of this from a rehabilitation point of view. And that's what we're kind of working at right now. One of the things that was really exciting, and I mentioned it subtly before, is that a lot of the action observation based approaches have been merited or somehow associated with the finding of mirror neurons, and thinking that mirror neurons are what's driving these representations of how to perform movements. But it may not be as simple as observation alone, but maybe how action is being observed. And what parts of the visual scene are you grabbing together, and how is that helping to support parietal frontal activity in the brain? And so gaze may be a powerful tool in modulating this parietal frontal activity during action observation. And some of our preliminary data that I haven't showed here seems to suggest that that's exactly what's happening. And it seems like it's happening based on an implicit strategy development for motor control and motor learning. And that while you're looking at these behaviors, you're implicitly extracting out the information that's going to help support your performance. It's very similar to what we think we're seeing even with those static images of tool objects in context with the hand. As gaze is differently organized about that, that is a process that we did not ask the participants to do. This is something that just emerged. It developed as they were gathering information about the perception of the tools and actions that they were looking at. And so this leads to a question that we've been looking at for several years now, and we're starting to begin to understand a little bit more. Does gaze change, and maybe just gaze change alone, help shape parietal frontal activity to prime motor learning? Is it possible that during these action observation-based approaches, whether it's in a rehabilitative sense or in a basic motor learning or development sense, that those experiences alone, just the observation phase, is enough to shape these parietal frontal networks? So there are several studies that are starting to emerge and also showing very similar findings, looking at the ability, for example, to observe, let's say, a simple movement like this, which may activate or cause corticospinal activation differences for the muscles that are involved in that movement, but not the muscles involved in this movement, right? And so the idea here is that even at a corticospinal level, you can prime the motor system based on the specifics of what you're looking at, not the general sense that you're looking at something. So in the case of upper limb motor control and prosthesis, there's been a lot of work looking at motor learning to support prosthesis use, and also a lot of work in developing more advanced bionic prostheses to support usability and adaptation, which are all exceedingly great. But maybe we've been missing out on the potential of another sensory system, the visual system, to actually support functional learning with prosthesis early in amputation therapy. And so that's kind of where a lot of our work is going now, to try to understand some of those processes. And so just by way of conclusion, the good old wall of text here, it seems as though we have that gaze behavior, can help predict judgments related to action goals. We can actually run a lot of those studies that I mentioned earlier in the talk to be able to actually predict exactly what image people are looking at. These gaze patterns are incredibly robust to prediction. But it's possible that we can predict these goals and that somehow these parietal frontal mechanisms are involved in doing that. And this activity may be influenced or influence gaze behavior. We're trying to understand that directional coupling right now. and particularly how it may be useful for neurorehabilitation approaches to really help to understand what's driving these changes in these circuits. And so something that's very important to me, acknowledging the good collaborators that I have, I mentioned particularly in the development of action encoding, Anna Borgi, who's now in Rome, who does do a lot of work with her related to this as well. Studies that we've been looking at with development of motor development and action syntax, looking at that from the lens of anthropology, which is just a lot of fun. I haven't talked about that, but I'd be more than happy to share that at another time as that matures a little bit more. We start to get publications out. And also looking at the work in amputees and motor learning between some good colleagues in mechanical engineering, in the clinical sector, and also at Georgia State in those collaborations. So with that, I will end. Thanks so much for the attention and happy to answer any questions.
Great. So there's lots of virtual applause going on. Another disadvantage of this framework is you can't hear them all, but they are there. We've had very good turnout for this seminar series. Thanks all for continuing to participate. What we're going to do now, we have plenty of time for questions. Again, I would like to prioritize this question from a student or a postdoc first, but while we're doing that, I'm going to have people can either write their questions into chat or they can raise their hand with the little raise hand function, and I'll call on them. But before we go to the questions, and while people are writing those out, there's a quick announcement from Lena Ting about a workshop that's coming on. So apologies, Louis, for hijacking your presentation, but I'll give some time for people to formulate their questions. And I'm going to share my screen really quickly, and Lena is going to present this really fast. Go ahead, Lena.
Great. So thanks, everyone. You may have seen this announcement, so I just wanted to remind everybody we have a workshop on human and animal movement. Where does stereotypy end and variability start? Thursday, March 18th from 10 a.m. to 1 p.m. The registration, I've posted it in the link. We also have, in addition to the speakers listed here, Mackenzie Mathis will also be speaking. And this will be 10-minute talk, 10-minute discussion. 2 days later, once you've attended that, we'll put that into action. If you want to learn how to use Deep Lab Cut to do video-based movement analysis from start to finish, that will be a Saturday 9 to 3 sort of event. You don't have to have any experience with any of this, and we'll provide you with lunch and put you in teams and give you some cool videos. We need you to sign up by Monday, March 8th because of the logistics of all of this. So I put that in the chat. So thank you.
Great, thanks, Lena. Louis, if you want to go ahead and share your slides again, you can go ahead and do that. Now I had to take control while it was on. We've got some questions starting to come. We got some comments from Lena has put some comments into the chat box. So, and we'll go ahead and take some questions. So we're having some resistant questioning. So again, oh good, we've got some questions. So I'm not sure if everyone is, what everyone's role is. So Isha, I'm not sure if what your position is. If you're a student or a postdoc, please go ahead and ask your question. Otherwise, hold on for just a minute. We'll give some priority to those. You feel free to unmute though.
Hi, I'm an undergraduate student, and I just
Perfect.
wanted to ask a clarifying question. When you were discussing the effect of the changes in gaze alone on motor rehabilitation, I just wanted to clarify, would the correlation between the increased gazing at the shoulder improving the peak shoulder velocity? Oh, wait, sorry. Sorry about that. Would the correlation between the increased gazing at the shoulder improving the peak shoulder velocity be an example of the gaze alone improving rehabilitation?
Yeah, good question, potentially, right? So, one of the things that was really the increased attention to the shoulders correlated to the overall movement velocity increasing. So their ability to kind of grab and reach across. And, that's one of the things that we could speculate. We saw that effect very early on in the first block. So before the user had ever any performance with that task, in the first block, you saw that improvement already. And being able to look at that effect, even at the earliest stages of therapy, would tend to suggest that perhaps the observation alone is sufficient. But we need to understand more about, other experience-based factors and things like that that could lead to this.
Okay, thank you. And also
Mhm.
congratulations on your grant proposal.
Thanks. Hopefully we get it. That's a win for all of us.
And Isha, thank you for volunteering. You made a lot of faculty members very relieved because now they can start getting in the queue to ask questions. So we really needed that going on. So appreciate you launching us with the first question. Our next question comes from Dolly. Dolly, if you want to unmute and ask your question, feel free.
Hi, Louis. Dolly here. I took your class last semester, so super excited to see your research. So, this talk seems like you're trying to parsing out like eye movement and also strategic planning in some ways, like motor control. So have you thought about maybe doing some kind of like instructions at the start to kind of cue participants to look at shoulders and see whether that changes the way that they plan beforehand? Beforehand. Unintended.
No,
So.
yeah, absolutely. That was one of the more exciting things about this study was the realization that these patterns were just emerging. We weren't telling them to look at anything. And I don't think I mentioned that in the talk. We just told them to watch the video or watch the action and copy it. But certainly there are some people that, for whatever reason, they don't look at the shoulders or they don't look at certain things. And yeah, you're right. There could be a way to, take that which has so far been implicitly driven, but to make it more explicit now in the instructions. If somebody isn't compliant, what would it be like to say, all right, look at the shoulders, or maybe highlight the shoulders in real time if we can observe in real time that their eyes aren't looking there. We could do some visual cueing to orient where they should go. And certainly that's something that we've thought a lot about developing into some of our studies, and is actually part of our collaboration with Georgia State right now.
Fantastic. Good. We're getting more questions. So feel free again to raise your hand or put your questions in the chat. Louis, I'll ask a question. So there's this great workshop apparently coming on that asks the question of where does stereotypy end and where does variability begin? I think that's a great question for your stuff, meaning like how consistent is all of this? What drives variation? How important is variation in this? I mean, like, how does that play in? You must have been thinking about these questions a little bit.
Absolutely. And, really one of the areas that this goes back to in the case of, and I'll just put it up there again, I'll go back to this one here. So I skipped this particular slide where we were trying to understand with very discrete kinematics with the elbow flexion, extension, and the shoulder ABA deduction based on what the participants see, which is a very periodic same pattern because it's a recorded video. And what the prosthesis users do, one of the things we noticed was that, obviously there's some similarities between these two things, particularly at the level of the elbow, but it is a very regular pattern. All of the participants uniformly just automatically reflects into this behavior, which really got us intrigued at wondering, okay, why is it the case that people just kind of automatically emulate in this way what they're seeing? Because when we first start them out in the study, in a separate study, and I don't have slides on this one, but in a separate study, we actually looked at just their basic variability. So when they're reaching for things and doing things, it's just kind of, there are certain patterns that are constrained, and in part because, they don't have hand movement, they don't have wrist movement, they don't have forearm rotation. It's all prosthesis use at the level of the mid forearm and up. So they have their elbow, they have their shoulder. Some things are constrained, but some things are free to vary. And within that, there's a lot of variability in terms of how people will just naturally move when we have them do the same types of tasks. So, really, what happens when we, over time, we take them away from this setting and then just have them do tasks over and over again? Do we see the emergence of stability? Do we see that kind of emergence of chaos, so to speak, or irregularity again? Or do certain joints begin to hold? It seems like, honestly, the result is highly variable, and that's exciting for us. That's something that we're definitely looking at in some other work to understand how that variability relates to the motor processing.
That's really cool. I guess the other thing that I was sort of curious about on the variability side of things is going back to the very beginning. This might just be my naivete about tool use studies, and so maybe this is a well-known phenomenon, but you had the scoop and the ice cream in that example, and that's something that we have a strong association with the correct tool for that. But occasionally, like, I run out of clean spoons, and I'm like, well, I've got a knife, and I can sort of use that, but that's not something that I normally associate. I was sort of curious, like, how does this extend, especially this relationship between different brain regions, to whether the object is always been associated with that particular function and that particular use, or when we're doing that sort of more abstract or interpretive connection between things? I don't quite know what the right words are. Do we see the same kinds of patterns? Is that known?
Yeah, that's a great question because it harkens on a lot of work that we've done over the past probably about 10, 15 years. So we actually have done a good bit of work related to, I don't think I have it up here in this particular set here, but it relates a little bit to this study here. But we've looked at this sense of what it means to be spatial, or what it means to be in reasonable proximity. So if we actually developed a theoretical model around the idea of what happens if I show you a knife and a coffee cup. Yeah, they don't typically go together automatically, but you could certainly think about stirring with a knife just like you could with a spoon. And so we've actually studied that from a neurophysiological point of view using EEG and fMRI a lot. And we do see actually a lot of the same left hemispheric parietal frontal circuits engaged when you're seeing that. But at some point, you start to see neural areas that more relate to violation of action. And that's kind of what's shown in this image here in green. These are areas that if the action that you're seeing is just absolutely wrong, now you see a lot more temporal activation into the insula, which has actually been seen by several researchers as well. So in that gray space, or in that creative space, I think as we called it in our theoretical model, there's kind of still this parietal frontal network heavily engaged, maybe to a greater extent.
All right. So it's gone on to about 12:15 now, so I think we'll go ahead and wrap up and thank Lewis again for a really wonderful talk. I enjoyed learning about this stuff. And for everyone else, we will continue to have our neuroseminar series every Monday. We have one small change to the schedule. On April 5th, we had a cancellation, actually a postponement, but fortunately we have a new neuroscientist in the Department of Math here, Hannah Choi, who will be giving a talk about her models of networks and network theory as it relates to vision. So she'll be giving a really nice seminar on April 5th. So thanks, everyone. We'll see you again next week. And thank you, Lewis. That was wonderful.